Hello, everyone. First of all, I want to thank Professor Andrew Adamatsky for inviting me to contribute to this interesting workshop on the advances in unconventional computing and giving me the opportunity to present my talk entitled An Unconventional Contribution to Quantum Artificial Intelligence Based on Chemical AI. In my presentation, I want to answer two fundamental questions. First, what is chemical AI and why am I developing chemical AI? And then I will present an achievement of chemical AI that is an unconventional contribution to quantum AI based on quantum states encoding fuzzy information that allow us to devise biologically inspired photochromic fuzzy logic system to extend human vision into the UV spectrum. Finally, I will conclude by presenting some perspectives. Let's get started. What is chemical AI? Chemical AI sprouts from natural computing. That is an interdisciplinary research line, drawing inspiration from natural phenomena to formulate new algorithms, propose new materials and architectures to compute, and new methods and models to understand complex systems, based on the rationale that any distinguishable physicochemical state of matter and energy can be used to encode information. And every natural transformation of these states is a kind of computation. Within natural computing, we distinguish two programs. In the first, scientists exploit the physicochemical laws to make computation. In the second program, scientists mimic the information gathering and utilizing systems that belong to living beings. Chemical AI derives from both programs and is trying to answer the following heuristic question. Which biological intelligent competencies can be mimicked through inanimate chemical systems in wetware, that means in a liquid solutions, which is the characteristic phase of life? Traditionally, artificial intelligence is developed through two strategies. One strategy relies on software running on current electronic computers or special purpose hardware. Such software either reproduces the human thinking process when it is a flow or rigorous logical operations, or it mimics some structural and functional features of neural networks to learn how to perform tasks from data, or it mimics some other natural rational phenomena, for instance, biological evolution. The other traditional strategy to develop AI relies on reverse engineering neurons and their networks in hardware for neuroprosthesis or to design pre-light computing machines, revolutionizing the von Neumann architecture of current electronic computers. Recently, a brand new strategy to develop AI has been proposed is the so-called chemical artificial intelligence that relies on molecules and macromolecules that operate as if they were hardware, but they are dissolved in liquid solutions, where phenomena such as chemical reactions, chemical waves, diffusion, migration, advection, convection can occur, and these phenomena operate as if they were software. In other words, the hardware and software of conventional AI are merged in the vector of reactive chemical solutions, maintained in out-of-equilibrium conditions. Here I report a provisional map of chemical AI, where we distinguish three main avenues. In the first, scientists, chemists mimic the human thinking process by developing chemical logic gates and Turing machines. The second avenue is neuromorphic engineering in wetware, and the third and most recent avenue is quantum AI, where chemists mimic the phenomena revealed in the field of quantum biology, and they propose alternative strategies to develop quantum law. Here I report the knowledge map of chemical AI, where that is a three-dimensional graph projected on a plane. Along the x-axis, we have the chi domains that are molecular and supramolecular chemistry, system chemistry, including micro and nanofluidics, synthetic biology, and finally multiscale chemistry. With multiscale chemistry, I refer to all those chemical systems that manifest self-organization at different spatial scales and multi-level causality when appropriate macroscopic thermodynamic forces are applied. Multiscale chemistry is required to mimic some structural and functional performances of multicellular biological organisms, exhibiting multiscale competencies. In the knowledge map of chemical AI, along the y-axis, we have the Chi paradigms that are all the biosensors, brains, and effectors. And finally, in the third dimensions, represented by the saturation degree of the green color, we have the biological intelligent competencies that range from sensing to perceiving, from computing to planning, from working to acting. Some of these competencies have already been implemented. They are indicated in black. Some others have not implemented yet. They are indicated in white. Probably we require a further development of system chemistry and multiscale chemistry to make these competencies be within reach, or probably they cannot implement it through inanimate chemical systems in weather. Because if we think deeply about the two sets of competencies, that in black and that in white, we discover that they are separated by a still unexplained concept, that of consciousness. Why am I developing chemical AI? Well, an ambitious goal of chemical AI is the development of chemical robots. A chemical robot is envisioned as a confined, complex molecular assembly that reacts autonomously to its environment because it has sensors collecting data about the environment and the internal state of the robot. It has artificial neural networks processing the sensory data, making decisions, and triggering the actions of the effector modules that can affect the surrounding environment. The intelligent activities of a chemical robot should be sustained by an appropriate metabolic model. Chemical robots should be easily miniaturized in order to be implanted in living beings and interplay with cells and organelles to perform biomedical actions. In other words, chemical robots should become auxiliary elements of our immune systems. In this case, lymphocytes are the best prototypes for chemical robots. But we might think of deploying chemical robots in the environment to fight pollutions or face problems related to energy and food supplies. In this latter case, microorganisms are the best prototypes for chemical robots. So it's evident that chemical robotics is a new avenue in the area of robotics, because chemical robots are completely different from traditional hard robots. They are somehow related to soft robots, although they are expected to be truly tiny. The development of Kai might have a curious side effect. It will probably assist human efforts to unveil the mysterious event of the appearance of life on Earth. That staggering event was described as a phase transition or sudden change from a completely abiotic world to a world where the first complex chemical systems, capable of exploiting matter and energy to encode, collect, store, process, and transfer information to pursue goals, somehow pop up. And now, after answering these two fundamental questions, I present an achievement of chemical AI that is an unconventional chemical contribution to quantum AI based on quantum mixed states encoding fuzzy information that allow us to devise biologically inspired photochromic fuzzy logic system to extend human vision into the UV spectrum. First of all, quantum AI is a recent research area derived by merging quantum information science and artificial intelligence and expecting a positive feedback actions. The quantum information science can speed up artificial intelligence. On the other hand, artificial intelligence can support quantum computing, for instance, in error mitigation and quantum circuits optimization. Conventional quantum information science relies on qubits that are superimposed quantum states. Our contribution to quantum AI is unconventional because we rely on thermalized quantum mixed states implementing molecular fuzzy sets. So first of all, it's worthwhile reminding some basic idea of fuzzy logic that has been defined as a rigorous logic of vague reasoning because it reproduces the exclusive human features of making decisions using deductive reasoning based on qualitative information expressed through the words of the natural language. Any nonlinear cause-and-effect relationship involving input and output variables is described by building a fuzzy logic system that relies on syllogistic statements of the type if-then, called fuzzy rules. The fuzzy rules use adjectives for the input and output variables. Adjectives are fuzzy sets that granulate and graduate the numerical values of both the input and output variables. A fuzzy set is different from a classical Boolean set because an item can belong to a fuzzy set with a certain degree, which can be any real number between 0 and 1. Here, as an example, I report the granulation and graduation of this temperature range in 4 fuzzy sets, but the number, position, shape, and graduation of the fuzzy sets are context-dependent features. Here, in this picture, I report the schematic structure of a fuzzy logic system where we distinguish 3 elements. The fuzzifier that transform crisp input values in degrees of membership to the input fuzzy sets, the fuzzy inference engine that transform the activated input fuzzy sets in specific output fuzzy sets, and finally, the defuzzifier that transform the activated output fuzzy sets in a crisp output value used for a prediction or for making decisions related to the phenomenon that the fuzzy logic system describes. One of the most iconic successes of fuzzy logic is its capacity to control the remarkably unstable inverted double pendulum. And since in certain circumstances, especially in the wild, living can be imagined as unstable as the state of the inverted double pendulum, well, my thesis is that any living being succeeds in surviving because it processes fuzzy logic at different spatial scales, ranging from the molecular to the organismic level. And today, I want to demonstrate how fuzzy logic is processed at the molecular level. Microheterogeneous chemical systems implement molecular fuzzy sets. What does it mean? When a chemical compound exists as a collection of distinct conformers and/or experiences microenvironments with distinct physicochemical features, it works as a molecular fuzzy set because it has context-dependent features and it granulates physicochemical variables. And now I'm giving you a demonstration of this definition. First of all, I exploit this merosaline that is a pretty flexible structure because it can exist in many conformers. These molecules, the structure that you see, have the same molecular and structural formulas. However, they differ in how the atoms are oriented in the three-dimensional space. They are conformers. A merosaline is produced by UV radiation of a spiroxazine. That is a direct thermally reversible photochromic compound. The structural change is accompanied with a significant spectral modification. Spiroxazine is uncolored, merosaline also absorbed in the visible region. If we record how the absorbance in the visible changes over time, we detect an exponential growth when the system is upon a stationary UV radiation. And as soon as we discontinue the radiation, we have a recovery of the original state because the merosaline is a metastable structure, so it transforms back to the original spiral form. Since the merosaline can exist in many conformers, it's reasonable to use a polyexponential function to fit the bleaching kinetics. And I use the maximum entropy method for its determination because its output is the least biased estimate possible on the experimental data. Here I report some results for the photochromic compound dissolved in pure ethanol at different temperatures. And we see that the higher the temperature, the broader the distributions of the lifetime and the shorter the lifetimes. We can affect this lifetime distribution through chemicals. For instance, injecting glycine. And glycine exerts a double effect. It shrinks the distributions and it lengthens the lifetimes. This double effect is probably due to a supramolecular interaction between glycine and the zwitterionic forms of the merosaline. So it's evident that the lifetime distribution is associated with a conformational distribution of the merosaline. The shape and position of this conformational distribution depend on the physicochemical context. This conformational distribution granulate specific portion of the variable time. So it's evident that this conformational distribution of the merosaline has the features of any molecular fuzzy sets. But since each conformer is represented, described by a peculiar wave function, the conformational distribution is also a quantum state. In this definition, Wi prime represents the probability of the ith wave function, but also the degree of membership of the ith wave function of the ith conformer to the molecular fuzzy set. And the amount of information encoded by this conformational distribution is given by its fuzzy entropy measured in fits. When we manipulate molecular fuzzy sets, we are actually processing fuzzy information that is an unconventional form of quantum parallelism. The temperature allows the decompression of fits, so it's a decoder. Glycine allows the compression of fits, so it's like an encoder. We can increase the degree of computational parallelism by preparing mixture of distinct quantum states. That means a mixture of distinct molecular fuzzy sets. And for the chemical composition of these mixtures, we should be driven by our attempts at granulating the physicochemical variables. In this regard, a remarkable example is offered by human color vision. In the center of the retina, we have three types of cones that have three distinct photoreceptor proteins, the so-called red, green, and blue proteins. They share the same chromophore, that is the photochromic 11 cis retinal. However, the three types of proteins differ in the position of the lowest energy absorption bands because they differ in the amino acid composition of the pocket embedding the retina. These three absorption bands are associated with distinct transition moment integrals. They are characterized by peculiar vibronic wave functions. And these three absorption bands are three molecular fuzzy sets granulating the visible region. Lights having distinct spectral composition belong to these three molecular fuzzy sets at different degrees, and the collection of different degrees is interpreted as a peculiar hue within our brain. The granulation of the visible region is a form of quantum parallelism that enables humans to distinguish around 200 hues. So starting from the premises that the granulation of the visible region allows humans to distinguish colors and direct thermally reversible photochromic compounds absorb the UV and become color, we ask ourselves if it is possible to discriminate the three UV regions exploiting the hue of color generated by a photochromic system. The answer is that one photochromic compound is not enough because the position of the band generated in the visible region by one photochromic compound is always the same, whatever is the excitatory wavelength in the UV. But we can prepare a mixture of photochromic compounds. And the selection of the chemical composition of this mixture should be fixed considering the granulation of the UV regions through the absorption bands of the uncolor forms and the granulation of the visible spectrum through the absorption bands of the color forms. Here I report a fuzzy logic system based on these quaternary solutions, photochromic solutions. The fuzzifier is given by the absorption bands of the uncolor forms. The fuzzy inference engine is based on the photochromism of the four compounds selected. And finally, the defuzzifier is the human eye that perceives the green color, the gray color, and the orange color whenever the photochromic system is irradiated by frequencies belonging to the UVA, UVB, and UVC, respectively. So this is an example of a biologically inspired photochromic fuzzy logic system that extends human vision into the UV spectrum. And finally, I'll move to the conclusions by presenting some perspectives for chemical AI that refers to the unconventional strategy for mimicking biological intelligence in web. First of all, chemical AI and chemical robotics will help humans explore and colonize a pool investigative space of art, that is the molecular world. The colonization of the molecular world will provide tools to effectively combat poverty, clean up the environment, overcome diseases, and extend human longevity. Chemical AI will also contribute to the development of quantum information science by devising new strategies for sensing, computing, and communicating through quantum states. Finally, trying to obtain intelligent chemical systems from scratch can help understand intelligence and life. I want to acknowledge my current collaborators, my young resources, the European Union and my university for the financial support, and all of you for your attention. You can follow me on the social media, and I invite you to contribute to a special issue on chemical AI open to submission in this MADPI journal. Thank you for your attention.